You found the skeleton. How would you tell people that this happened? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
We also have a guest on today by the name of Monk Man. Welcome to the show. Glad to have you.
Yeah, thanks for having me.
So, you and I were talking on Twitter, and I don't remember what kicked our conversation off originally, but we decided in DMs on Twitter to discuss speciation, the implications of speciation, and how far descent goes. Does that sound fair?
Yes, for sure.
Okay, so that nobody here strawmans you or misinterprets your position, misunderstands, or anything like that, would you please tell us what you think about speciation and about how far descent goes in your own words?
Sure. I would kind of take the classical creationist position and say a species, I would call, I would start at the family level of the animal category, and say that those are the any breedable kind is a distinct species that stays the same species throughout time. And so, yeah, I would say family is, I suppose, my level or equivalent maybe to what you would call species. But there are some exceptions for that for certain animals, and maybe we can get into some of those.
Could I ask for a clarification?
Yes.
Sure.
What do you mean by family is your species? What do you mean by that?
Well, that was a different one, but yeah, that's a good one too.
Oh, I'm sorry.
I guess I would say family for me is a kind of animal when we talk about I'm not totally familiar with higher terms and stuff in the science world, so I'm
That's
trying
fine.
to so a family, so like a canine would be the family of a classification of animal that they can only breed with each other, and that's what they continue on as.
Okay.
So,
go ahead.
So the question that I want for clarification is, you said that a species had to be breedable, and I just want to know what exactly that means, because there's a spectrum of breeding compatibility among extant animals. So what exactly do you mean by that?
So so as long as they can interbreed with each other, then they are the same. They are the same family.
Okay.
Okay.
I have a follow-on question, which is, what constitutes breeding together? Like,
able to have offspring, and that could be fertile or infertile. But then that also that's also not the definition either, because of course there are branches that can't interbreed any longer, and I recognize that as well. So that would kind of be an exception to the rule, but it would still be it wouldn't place them outside of that family.
So if two organisms can create offspring, then they are in the same species according to your definition. But if they can't, they still might be.
If they so if they can't, and they still have all, that's where I would refer to the taxonomic features. I mean, that has to kind of be the foundation, obviously, of defining a family kind.
Well, that depends on who you ask, because there are lots of different definitions for species. Family is even more arbitrary, and so is genus, than species is. The species is really the only level within taxonomy that you can assign like an objective definition to. Now, it may not be a totally correct definition. There may be exceptions, but that's the only level you can assign a definitive definition to. All right.
Well, let me.
Oh.
I also want to get a handle on what we're talking about with families. So, your contention is, you gave it as an example. So, Canidae would be an example of what for you would be a grouping that you would consider to be a species under your, I would say, somewhat broader definition. Which I mean, it's fine to have a stipulative definition, so that's fine. So, would you say that, say, foxes, African painted dogs, maned wolves, the gray wolf, jackals, raccoon dogs, and all those are all descended from an original population that was themselves a group of canids?
Yes, correct.
Would that also include the non-canine canids like the borophagines?
Would you mind explaining exactly that species? I'm not familiar.
So Borophaginae is a now extinct subfamily of Canidae, and they were generally larger, more robust, and had teeth specialized for bone crushing, similar morphologically, in gross morphology, to like modern hyenas. And basically, we only know them from fossils, but they're traditionally placed within Canidae as the sister subfamily to Caninae, which is the group that includes all extant canines.
Okay. Yeah,
So
I would have to. Oh, sorry. Go ahead.
If you don't know that one, then I mean that's fine. If you've never heard of borophagines, I can't expect you to have a position on them.
Yeah. Well, it sounds kind of similar. I mean, I guess to a hyena, in the respect that if we were to find that fossilized and not have it alive today, it would be it would look like it was in the canine family. And so,
well, hyenas have been known to be more closely related to cats because of like dental morphology and other anatomical characters.
EA for scent marking like a cat.
Yeah.
right. Yeah, I know they have their own distinct family. Actually, not.
Yeah,
They're
Hyenidae with the
standalone.
Yeah. Okay, so it sounds like for the most part you're taking the same basic position on kinds or species, as you're referring to them, that like Answers in Genesis is basically taking. Where if you look at their purported kinds list, it's mostly a list of taxonomic families.
Mm-hmm. Yeah,
Okay.
That would be more or less.
Okay. Now, my concern with basically using the family level as a proxy for where you're going to draw a line with common descent, even in general, because you said that there are exceptions with that. I mean, that's fair, right? But my concern there is that fundamentally, family doesn't mean anything, right? A family is a genus is a group of similar species. How similar? There's no rule. And a family is just a group of similar genera. And how similar? Again, there's no real rule. So it's just as valid to say that rather than having we'll pick something other than canine, something with more close, relationships, at least in and as the science has determined it. So let's say we take like Arctoideans, right? So that's bears, procyonids, so that includes things like the raccoon, as well as a number of other South American carnivores, and pinnipeds, as well as mustelids and mephistids. So that's like weasels, bears, skunks, raccoons, and seals, and also sea otters. Otters generally.
Yeah.
So in this, from what we've generally been able to determine, bears and pinnipeds, say, are very close related in terms of evolutionary biology. And there's no particular reason taxonomically why those two couldn't be a single family. We just traditionally haven't said it. So I guess my question would be, other than just the fact that the names end in IDAE, like, pinniped or pinnipede and ursidae, what actually makes it so that you think that those groups don't share common ancestry? Because it can't just, I guess it could be. It doesn't seem internally coherent to say that all that really matters is that they've been traditionally placed into these ranks called families because that's a fundamentally arbitrary thing, right?
Right.
So what is it about, say, bears and seals, or bears and raccoons, or raccoons and skunks, or even skunks and weasels, that for you says, no, this is a bridge too far?
That would be simply the body structure.
What
I mean,
specifically?
Yeah, what like if I said okay, so we have the Procyon, right, is the genus of the North American raccoon, right? And then we have like Ursus, which is the genus for like I think it's like black and brown bears and polar bears, if I remember correctly. What specifically prevents a raccoon from being related to a bear in terms of its actual morphology? Is it like something about the teeth? Is it something about the orbits, the axial skeleton? Like, what are we talking here?
well, I would say paws for one. I mean, I'm not a let's see, biologist in that regard, but the hands are certainly different than a bear's. They can grip. The thumbs, right, are opposable like ours. So
Well,
that's a
they're not
huge
quite
genetic
as
difference.
opposable as they're not quite as opposable as human thumbs, but they are certainly more flexible than bear.
Okay.
Than bear paws. Paws is probably the right thing. But I mean,
Yeah.
fundamentally, though, raccoons and bears have the same number of fingers with the same number of bones in each one. They both walk on their palms and heels, like sort of like humans who walk on their heels. I mean, is it just that raccoons have a wider range of motion for the digits in their hands?
I mean, that could certainly be one. I mean, tail is different. I'm just kind of thinking off the top of my head.
I mean, the tail is different primarily in being longer.
Size, right?
Canidae also has a wide range in tail length, but I don't think that is on its own enough for you to say that therefore Canidae represents more than one fundamental created unit.
Right. Yeah, that's definitely a closer morphology example for sure. I'd have to take a closer look at it, but yeah.
So are you saying that you would be open to, say, Procyonidae and Ursidae sharing common ancestry?
It would be cause for further investigation for sure.
Okay.
If I'm a scientist, I would have to. Yeah, at first glance, I couldn't say, yeah, these are definitely two separate families. I would have to, really dissect and study the features and see what is, because obviously there's a lot that look the same, but then when you actually take a closer look under the hood, it's really subtleties that are actually very key to being separated.
Yeah. Well, that's one of the things that in science, one of the ways that taxonomy tends to work is that the same reasons that we say that, say, the members of the same genus, right? So to take your example with canines, one of the most widespread genera in Canidae is the genus Lupus, right? So you've got the wolf, you've got the golden jackal, you've got the coyote. These are all very widespread animals, all in the same genus, right? And the same reasons that science says that these are related also extends to the rest of Canidae, right? So like that's also why we say that like the red fox, the fennec fox, the maned wolf, the painted dog, the dhole, etc., are also all related within Canidae. But then similar morphological reasons, at least, since now we're getting to extinct things, is also why we say that they're related to the borophagines, the bone-crushing dogs. But also, very significantly, those are the same reasons that we say that they're related to even more distant things like Hesperocyon or bears or seals, or even more distantly, cats. So I guess my question is, how do you know where to cut that off? Because you said that breeding means that they are in the same group, but not breeding isn't necessarily evidence that they aren't. So we can't just say, well, because dogs and bears can't interbreed, they can't be related, because we already know that not breeding isn't evidence against common ancestry. So is there do you have some amount of difference or some kind of criterion that you can articulate in a way that, you know, would be able to be broadly applied to any group of animals that actually shows you where the cutoff is? Or is it sort of you kind of eyeball it and this just kind of seems like what it should be?
I would say we have to define what a feature is and really zero down on function and specificity and what if is there a difference in function from these two animals, and that trying to think, so the raccoon and the bear, yeah, like I mean the nose, I would imagine, has to be and the raccoon a lot less powerful, so that would be
I'm actually not aware of any indication that raccoons and bears have significantly different like levels of ability to smell. Do you have anything that suggests that? Because I've never encountered that. I'm open to it. I just have never heard it. Do you have anything that actually indicates
I
that?
could be I could be totally wrong. I'm going off of kind of the bear's nose being this being like one of the strongest in all like the animal kingdom, but raccoons could be similar. I'm actually not sure.
Well,
but we'll go back to the paws then, I guess.
Yeah, sure. Okay.
Yeah, that thumb is a very, or opposable appendage, whatever you want to call it, is a very distinct, has a very distinct function, right? That the bear can't replicate, and that's something that we can classify. We can draw a model of it. We can observe it performing actions, whether it's cleaning itself, gathering food, fighting, grooming, whatever it may be. That's a separation, and that would go into mechanics of how it moves, and so those are kind of the markers that I would look at.
So I'm currently looking at Ilurid and Procyanid Manus anatomy, and I'm actually not seeing the thumb as particularly opposable. It seems like what's happening in both groups is that the fingers are relatively flexible, and so they're able to grab things sort of like if you ignored your thumb and you just like, you could still grab like a can of soda or something without your thumb. And in fact, in Iluridae, there's a carpal bone that then extends to become opposable with at least digits 2 and 3, I believe. So like, I'm actually not seeing this significant difference in Manus anatomy other than that in Procyonids and Ilurids, which, in terms of science, Iluridae, which is the red panda and its extinct relatives, is the sister family to Procyonidae. So I'm actually not seeing, other than just longer fingers proportionally, what the big difference is between the Manus of a Ursid and the Manus of a Procyanid or Ilurid.
Yeah, well, that may be the case. I'm not. I wouldn't say 100% that it's opposable in the same definition that ours is.
Well, because also, I mean, Ursids do use their mani for food manipulation in a cruder but analogous manner to what Procyonids do, right? Bears pick things up; they catch fish with their paws and then direct that into their mouth and things like that.
Sure. So yeah. There's still. I'd have to suppose that it's definitely I mean the motion has to, like you were saying, the flexibility is very distinct from the bear paw, and that
I mean it's distinct in terms of like degree, but it doesn't seem like it's distinct in some kind of fundamental way. It just seems like Ilurids have proportionally longer fingers, which allows them a greater range of motion. And I don't think that, I mean, if we're going to say that proportional difference is the big thing that distinguishes, kinds, then we're going to have to split up caninae, because if you look at, say, the maned wolf, it has ridiculously long stilt-like legs compared to, say, a raccoon dog, which has stubby little legs. So, like, does that mean that we have to now break up the subfamily caninae into more than one different kind because of this difference in proportion that allows for a significant difference in range of motion?
Right. Yeah, that shouldn't be that shouldn't be a part of the consideration.
Because it seems like right now we're struggling to find a reason to separate families into different kinds when they are what science would consider to be essentially like sister groups.
I see. Yeah, that
And that seems like a problem for the contention that you can roughly peg the different kinds at the family level, because it seems like fundamentally there's not a huge barrier between the different families if all that we're saying is that, like, you know, two very similar families are different primarily in like the proportion of the fingers or the limb bones. That doesn't seem like it's such a big barrier, since we know that within families those things vary considerably.
yeah, I think it's one of those examples that's very again very hard to separate and to classify, but you know, given that I don't know much about the morphology and the taxonomy, that I would have to look into it.
We're getting like very, very deep into the weeds.
Yes,
on
we
this
are.
one example, and I think from this one example, I think it's fair to say that we don't see a kind distinction, at least in this example. So let's try to bring it out a little bit.
Jackson, just a quick
So,
mention,
sort of
because I forgot to say this before we started.
Sure.
If we have questions from the chat, I'll put those in the private chat here, and you
Yeah, I
already
see it.
have one.
Yep. Yeah, I see it. So sort of getting this out more broadly. This was a question I had sort of from the beginning. Was you mentioned that hybridization is sort of what entails a kind, and that, but at the same time, there are instances where organisms within what you would consider a kind fail to hybridize. And I can give examples of that too, right? There are organisms that become like secondarily asexual within a sexually reproducing group. So obviously they're not reproducing with anything else, right? So
Right.
would they still be part of that kind, even though
Jackson's favorite arthropod,
it is my,
Procambarus virginalis.
it's true. I identify with it.
Because you plan on reproducing asexually sometime?
I do. I
Okay.
you know, I just clone all of my ova. That's what happens.
Good luck with all of that.
So many questions. No, anyway.
Nope, I
so-
don't have questions. I'm just going to say good luck and leave it.
No, I have questions now.
Okay.
So with that example, we have a population, and this happened in recent human history. I don't know how old
Like
you
the
are. but
90s, I believe.
Yeah, you're right. In the 90s, you have this population of crawfish, Procambarus phallax, which is called like the slough crawfish. So they reproduce sexually. It takes a male and a female to make a baby. But within this population, within the last 30 years or so,
God, that makes me feel old.
I'm sorry. I knew it was going to happen as soon as I said it. But, you know, within the last 30 years, you had a totally new species come out of this group, one which reproduces totally asexually. So it not only doesn't reproduce with the parental species, males from the parental species will actually attempt to mate with because the new species is all female. They'll attempt to mate with the females and they will be unsuccessful. Researchers can watch this happen. So does that...
Researchers are into some weird stuff.
They watch a lot of weird... Look, if you ever... Dapper, if you want to know about a really interesting experiment, ask me about what the frog's eyes tell the frog's brain. That's a really interesting one.
Okay.
It's kind of disturbing, but it's very interesting.
I'm just saying, don't look into a crawfish researcher's browser history.
Or mine, Bandicoot feet.
Well, you know.
If anybody's wondering about the Bandicoot feet thing, I'll tell you later. So with that, would you consider the new species, which is asexual and therefore unable to reproduce with the parental population, would that still fall within the same kind, even though they are totally unable to interbreed?
It would, yeah. I think the fact that it can't interbreed is a sign of some genetic breakdown, some incompatible. Oh, sorry.
Incompatibility, yes, I'll agree with that terminology. Breakdown, not so much. What's happening is the crawfish, the new species, duplicated its one of its sets of chromosomes, so it has 3 instead of 2. And when you try to put those together, what you get is a condition called aneuploidy. So basically, you get unequal crossing over of your chromosomes when you make gametes, and you end up with lots of problems for your offspring. So yes, so continue. Go ahead.
Yeah, I've wondered if that was due to a fixed mutation or what exactly caused those chromosome differences. But
yeah, it was a total duplication of one of the sets. Yeah.
Oh, okay, it was okay.
Mhm.
Yeah, so mutation wouldn't separate it into a different family.
Okay.
I would just kind of label that as a sort of an exception to the rule, and the fact that you're saying that the males actually still try to interbreed with the females, to me is a strong indication that they're very much acknowledging that each other is the same family, the same kind of animal. So it's
okay. So
kind of a magnet on opposite, trying to line up, and they can't still.
Sure. I see what you're saying. So given that, and the reason I wanted to make that example is piggybacking off of that with a couple of things. First, we can actually produce the offspring that we produce with hamsters gets further developmentally than these two species do. So human sperm can actually fertilize a hamster's ovum when the zona pellucida is removed, which is a little protein coat, which helps filter out things like wrong sperm. But when you remove it, you can actually, human sperm can cause a hamster ovum to start dividing. And it'll actually undergo several rounds of like a division before it finally breaks down. Right? So go ahead.
Oh.
So humans can interbreed with hamsters.
Yeah, so we produce an offspring that is not viable for very long, but it is viable for longer than these two species of crawfish, than their offspring is. And that's between us and hamsters.
That
so what would that say for your idea of kinds?
I'm floored. I never heard that.
Yeah, it actually happened
before.
It's relatively common, or it used to be. I don't know if it still is, but it used to happen with like fertility clinics. That's one way that you can tell if sperm is viable, is you can use it to trigger development in hamster eggs. So that was the thing that used to happen. I don't know if we still do it, but we used to do it. So
interesting. That's very skeptical. You said.
I mean, you can look it up.
Yeah,
at least
it was a technique for testing male fertility.
Yeah, it's out there, you know.
So the how far along does the egg go? You said.
Like I said, just a few rounds of duplication, of cellular duplication. So like you have your zygote, which then duplicates into 2, and then you have 4, 8, blah blah blah. Well, it undergoes just a few, not very many, but still a few.
So we're talking like 16 or 32 cells or something like that.
Right. Yeah. Very. Still, just a little tiny clump of cells. Not very far, but still, that's more developmentally than Procambarus phallax and Procambarus virginalis.
Okay. Very. Yeah. Something's going on. Whether I would classify that as a reproducible effect is I think it would be called into question. I would have to read that article and see what other biologists are saying that actually is, because
that's fair, but it does raise the question: where does interbreeding ability cut off, right? Because you can say, okay, well, what about like African and Asian elephants, right? Like we only have one known instance of a cross between Loxodonta africana and Elephas maximus, which is the African bush elephant and the Asian elephant. And the offspring was sickly and died while still a child. Does that count? for making Elephantidae a single kind? And if that counts, like how sickly and young does the termination have to be before it stops counting?
I see.
That's
Yeah.
it's sort of same thing that I was asking basically.
Yeah, I would be curious to see kind of the definition of the cell being fertilized. What exactly is defined classically
For
as
fertilization.
yeah.
So
If that
fertilization. So, are you familiar with the terms haploid and diploid?
no.
So for us animals, well, for most of us, there are exceptions, but for most of us, especially for us humans, and also probably ceratosaurs, haploid. Well, haploid in general means you have half your set of chromosomes, which for us means just one, because we have two sets of chromosomes in all of our cells, and I say that in quotes, but you know what I mean. We have two sets of
Standard
chromosomes
body
in
cell,
all of our. Yes, a standard body cell, except for red blood
Except for
cells,
red
even though
blood
they make up
cells
the
and
majority of our
gametes
cells,
and-
but screw those guys.
Yeah.
For most of our body cells, we have two sets. We have one set that came from our mother and one set that came from our father. So a sperm cell and an egg cell each only have one set of chromosomes. They are haploid. Now diploid, di means two, ploid refers to the number of chromosomal sets. So you have diploid is 2, triploid is 3, tetraploid 4, etc. So haploid cells are, so our sperm cells only have one set, and egg cells, the ova, only have one set. Now, when an egg cell is fertilized, the set of chromosomes in the sperm are transferred to the ovum, and then there are now two sets within the ovum. It has become diploid, and then that cell, now having both sets, starts undergoing division and development, and then can become a baby. So that's what. Yes, monoploid does actually also mean one. So haploid could mean, so if you're like hexaploid, I see Bent asked, if you're hexaploid, that means you have 6 sets of chromosomes. Haploid would mean 3, so triploid is haploid. Monoploid is a thing, monoploidy. It's just another term for haploid, but nobody really says monoploid. Just fun fact.
They should. We should bring it. I was going to say we should bring it back, but we should just bring it into circulation because I don't think it was ever there.
I've seen it like
down with
Artiodactyla, I don't think
up with monoploidy.
So yeah. So that's what fertilization entails. So with the hamster, human and hamster, you got your human one set from the dad, and then the one set from the mother, from the female hamster in the ovum, and then these are brought into the same cell, and then you can start having your divisions.
Yeah, so generally, if you start having actual divisions from the ovum, it's generally considered to be fertilized.
Right.
If after a sperm goes in there, then it actually induces further division. That's basically how you tell that something has been fertilized.
Right. And so our question then is, where along in the process is your sort of barrier, right? Because you have on the one hand this very, very minor, you have just a few divisions versus like, a whole offspring which is born but then dies shortly after birth. Where do we draw that line, and how could we know that?
I think you would have to say the will to breed and fertility, and let's see.
I mean, sturgeons and paddlefish have had the will to breed, and they even produced infertile but offspring nonetheless.
And this is a difference in order, so this is actually about the same difference as a human and a hamster.
Yeah.
Yeah.
Actually, it's more so genetically, but like in terms of taxonomy, because fundamentally the taxonomic ranks are arbitrary. Genetically, paddlefish and sturgeons are actually far more distant than humans and hamsters are, but they still manage to make viable offspring. Well,
Yeah.
viable in the sense that they, become adults eventually.
Right.
The fertilized egg would be, I think, a good starting point. I mean, the line for me, again, I'm still very skeptical as to saying that's what's going on in this example with the hamster and the human. If it is, I mean, I that's really that'd be fascinating, and
I mean,
I could be very mistaken, but to me it seems like a kind of a outlier case, and I'm not willing to chuck my entire definition
yeah,
for it.
here it is.
So
Here's an article, the hamster zona-free ovum test. Here's the Wikipedia.
okay,
that's
yeah,
the
post that
Wikipedia.
article. That would be.
I'm
There
going to
you
put
go. It's in
that
our
in
side
the
chat.
in the main chat too, so that if anyone in the side chat wants to check that out, they also can. So I'm just going to read out the first two sentences from the intro. The hamster zona-free ovum test, or hamster egg penetration test, or sometimes just hamster test, is an in vitro test used to study physiological profile of spermatozoa. The primary application of the test is to diagnose male infertility caused by sperm unable to penetrate the ova. The test has limited value due to the expense and high false negative rate. So in this, sperm are incubated with several hamster eggs. And this is, in humans, right? The practice is made licensable in the United Kingdom by the Human Fertilization Embryology Act of 1990. So, I mean, this is a thing that occurs. Now, granted, as it said, there are false negatives, which means that, the human sperm is not super compatible with the hamster ova, but it is at least fundamentally compatible enough that there are, in fact, positives that come out of this, where, yes, there is, in fact, a successful fertilization of the hamster ovum. I mean,
Can't we?
The Wikipedia is just wrong, which I guess you could go with.
I was going to say, can we do that with any embryo? Is that like I've heard about, like, I think maybe sheep or pig at some point we were doing that. Was that?
Can you fertilize any ovum? Not. I mean, so there has to be a degree of compatibility between the chromosomes that you're looking at. I'm not an expert on the genetics of them.
I'm sure that if you try to combine a human spermatozoan with like a chicken ovum, you're going to get nothing, because there has to be a level of alignability between the chromosomes, and that's one of the reasons why you can get mules, right? Like the chromosomal difference between donkeys and horses is basically down to one chromosome. So for the most part, all those chromosomes can align, and so the cells basically know what to do. Now, in terms of what science says, rodents are basically the sister group to primates. They're technically the sister group to primates plus colugos, but the colugos is a fairly small group. So I'm comfortable with just saying you can think of it as the sister group to primates, which means that there is a fair bit of similarity chromosomally, so that you can get enough alignment to undergo a few levels of division before the cell just kind of stops because it can't continue on to figure out how to develop embryologically. And I mean, I'm anthropomorphizing the little zygote, right? But at a certain point, that compatibility is going to be so distant that you're not even going to get the cell to divide anymore. In that case, I would say that you have failed to fertilize. So it's definitely not the case that you can get human sperm to fertilize anything. But if our conclusion is anything that can reproduce, even if the reproduction doesn't result in a healthy adult, because remember, if say sickly baby elephants count, then we have to. If there's no if the cutoff just is reproduction, that is reproduction. And so if that means that reproduction indicates the same kind or same fundamental created unit, that now suddenly means that the group of Euarchontoglires, so that's rodents, that's colugos, and all primates, humans included, now share common ancestry. So it's also lagomorphs too. So that includes the rabbits and hares and pica and things like that. So that would mean that humans, by that standard, are now definitively related to, by genetic descent, not just chimpanzees, but rats and bunny rabbits.
Yeah. Yeah. I agree with the fertilization cutoff in theory. Um, I,
So would you be
again,
comfortable
I
saying
would have to,
humans and rabbits share common ancestry?
if that fertilization is actually taking place, that's I'm still not convinced that that's what's happening.
Well, now
Um,
I'm actually curious what it would take to convince you, because, I mean,
I
generally
think
speaking, there aren't. Now, Wikipedia isn't, the greatest source ever, but generally speaking, there aren't Wikipedia articles about like medical tests that just don't exist or anything. Like that's not normally a thing, right?
right.
So what is your explanation for just the bare existence of this as a thing that occurs if you don't think that the fertilization occurs at all? Like, what's actually happening, or what do you think might actually be happening that's fooling scientists who specialize in fertility testing into thinking that they have had a positive test, which then actually correlates with positive fertility in men?
Because that's their whole job. It's
Right,
not like they're doing a bunch of other stuff. Like that's literally their only
Right.
job.
This
It's
is-
just doing this.
There are actual physicians whose job it is to make sure that, to figure out whether or not a certain person's sperm can fertilize hamster ova. Like that's a whole thing. So what are they actually doing in your estimation? As is it possibly?
I think when it comes to much of terminology in science, definitions are in the eye of the beholder, or interpretations, we'll say. Especially let's say what we classify as life in the womb. I mean, that's kind of a hot debate, right?
I don't
It's
even think we need to go there.
Yeah, I'm also not sure
We're
that
just
it is a
talking
hot
about we're just talking about division.
Mhm.
Like we know, based on these tests, that the sperm is penetrating the outer membrane of the ovum. It is in fact depositing its set of chromosomes, and it is triggering division to occur.
Yeah, that's
That's all that's happening here. That's all that's happening.
If you saw that with a human spermatozoon and a human ovum, right, which is just normally how you get babies, right? If you saw that happen, you would just call that a conception, even if, like most human conceptions, it did not come to term, because most human zygotes don't end up making it
Right.
to be live births. So, like, I don't see what the big distinction is here, and unless you're trying to say that no, in fact, what's actually happening in these labs is that all the scientists who are conducting this test are actually getting it wrong, which is a position you could take. But then I would ask how you come to that conclusion.
I don't know if they're saying it is that the duplication is fertilization, or it is approaching fertilization.
No, the duplication only happens after fertilization has occurred. Like
Right.
fertilization has to take place. The chromosome set has to be deposited in the ovum first, and then only once that occurs can the cell divide.
It's kind
Because
of like
haploid cells can only do mitosis, right? They can only clone themselves essentially.
I kind of think of it like a tea kettle. You ever used a stovetop tea kettle?
Yeah.
The kind that whistles? The whistling isn't technically the boiling, right? It's just how you can tell that the boiling has occurred. You can kind of think of the cell division as like the whistling and the boiling as like the fertilization. Technically, the cell division isn't itself the fertilization. However, it's what happens when fertilization has occurred. So if you get the cell division, just like if you get the whistling, the fertilization has occurred, just like the boiling has occurred. You don't need to watch the water bubbling if you can hear the whistling. Similarly, you don't necessarily have to zoom into the actual nucleus of this particular hamster ovum and watch the chromosomes interact. The division is what gives you the evidence of the fertilization in the first place, because that's basically how you can tell that things have been fertilized. That is the result of fertilization, is the beginning of embryological development in an ovum. Or I guess what was formerly an ovum and what is now a zygote.
Right.
Okay. Interesting. Yeah. Then if that's the case and it's truly fertilization, then I guess I would have to pivot and say, what is the viability of offspring is has to become the new definition.
Well, sure, but that's
And,
what we've been asking: is where do we draw that line?
Yeah. Is it after 32 cells? Is it after 64, 128? Does it have to have been born alive? Does it have to reach adulthood? Where are we in this? And what is the reasoning for making the cutoff where you cut off?
It's a good question. Definitely something I would have to get who's a specialist in that field, an embryologist, something like that, someone who really knows, these classifications and these markers of reproduction to say, this is yeah. So I guess I'd have to plead the fifth.
Can I give you the spoiler alert though on what happens when you do that?
Sure, go for it.
Over 99.9% of all of the people with actual expertise in these areas will just say, yeah, no, humans share common ancestry, not only with hamsters, but with jellyfish. So if you're going to appeal to expert, like consensus or expert opinion, you're going to have to abandon separate ancestry because the experts absolutely do not agree with separate ancestry. Now, true, you can find one or two, relatively speaking, and by that I mean like you can probably find a few hundred who would agree with you about separate ancestry and special creation. But like when you compare that to the group that don't, if you're just going to go defer to expert opinion, I mean, it doesn't work out in favor of special creation or separate ancestry. It definitively comes out in favor of common ancestry, not even just of humans and jellyfish, but like of humans and bacteria and mushrooms and pine trees.
And further, to put another point on this this bit about development, as we mentioned earlier, because family is arbitrary and species are too, but that's our spicy take.
They're slightly less arbitrary than families.
They're slightly less arbitrary, right? Yeah. With development, because families are arbitrary, the amount of development that occurs from family to family is necessarily going to be different because a family is an arbitrary construct. There are families that have a single species in them, families with just two species. Then you have like beetles, like rove beetle, which has like 160,000 species in it.
Hey, when it comes to monospecific families, I'm just going to say I already mentioned Iluridae, one of the most adorable families of mammals.
They are very adorable. Can confirm.
Yes.
But so there's no singular rate or amount of development or single amount of nucleotide differences or single amount of morphological differences that defines any taxonomic category, not even species. There is no single feature of nature you can appeal to that will give you like a clear-cut boundary between, what we consider species. And I say that as someone who is in the process of reading like the textbook on speciation by Jerry Coyne and H. Allen Orr, because they also say this, and they're the guys on this subject.
You think if we 3D modeled an entire species and fed it into an algorithm and try and see if the computer can identify one body plan that fits within that family, that it could do that, or that it could not do that?
That I'm sorry, a body plan that fits within a family? Well, a body, again, body plan is another arbitrary term. Some people will say body plans are like the set of, and I've heard this term, this actually used as a definition. A body plan is the characteristics that a phylum has. So like all chordates, so basically all vertebrates, everything with a backbone plus lanceolate and tunicates.
Yeah, tunicates, except for the pelagic ones. Those guys can all die.
Yeah.
Sorry, I have an irrational hatred of pelagic tunicates, Monk. So like, just ignore me when I go on about that.
It's like, let me tell you. So basically, there are taxonomists, and it's not so much anymore with the rise of cladistics, but there are taxonomists who would say a body plan is the set of characteristics that unites everything in a phylum. So yeah,
Right,
vertebrates.
but that's
plus-
fundamentally arbitrary.
Right, but again, you're exactly right. It's arbitrary because you could say, well, yeah, the having a post-anal tail and pharyngeal arches and whatever else is the body plan of chordates, but having all that plus vertebrae is the body plan of vertebrates.
Right,
And
or
having
you
all
could
that
go-
plus vertebrae and yeah, go ahead.
I was gonna say you can also go the other way. Like let's say
Right.
we go with like larger groupings. Like let's say we go with like Lophotrochozoa, right?
Right.
Well, I mean, there's numerous phyla under Lophotrochozoa, like Brachiopoda
Mollusca,
and mollusca. Yeah. So-
Annelida, yeah.
Right. So like Lophotrochozoa fundamentally means things that have, lophora, the little feeding instruments.
Well, they either have lophophores or what is it, Trochozoa larvae? Isn't that
Right.
it?
So like, is that a body plan? Or we could go with like Panarthropoda, right? So we have like shedding of the cuticle, or just having a cuticle that then later sheds. We have segmented body plans with paired limbs on each body segment, along with like paired organs inside. Like, you have like paired nephridia in each segment typically. So like, does that mean that Arthropoda and Tardigrada and Onychophora, which are 3 different phyla, but they all have the same body plan maybe, because they all have that same kind of cuticle, they all have the paired limbs, they all have the body segment with internal paired organs, especially in some cases nephridia. That's one of the whole nephrozoa things.
Yeah.
So like, fundamentally, body plan is another one of those arbitrary things. It's useful for humans to talk about sometimes, but only if you understand that it's fundamentally just an arbitrary label that humans stick on organisms to make them easier to talk about. It doesn't reflect a real thing in living organisms.
Right. We have a biologist in the side chat right now who is actually a Christian. His name is Cy Gart, and we like him a lot on this channel.
Hi, Cy.
And as he points out, there are always exceptions because this is biology, and he's right.
Yeah.
No matter what category or no matter what label we try to stick on something, there are going to be exceptions to it. And there's a tendency in biology to teach students the norm and then say, but here are the exceptions to that, which I think is kind of the wrong way to go about it. But anyway, it's the best you can do when you have students who are really just interested in getting an A and getting out of there. But that's kind of the point, right? Is there are always going to be exceptions to these things for whatever category we try to erect or whatever label we try to slap on something. There's going to be an exception, and in a lot of cases, there are exceptions in both directions, such that it becomes like impossible to be like, okay, well, we're going to cut the line right here. It's like, well, why there and not over there?
Yep.
You get these gradations. There's actually Nestlig, who's one of my editors, sent me a paper today on what's called the species continuum, which when you look at nature, you see organisms in different stages of speciation, right? You can see when you have very recently separated populations, which are nearly identical in all aspects, to species where you have some populations which maybe some researchers would call them different species, some wouldn't. Like I was reading about some these lizards recently. There's this thing sadly called the European common lizard, Zootoca vivipara.
What a terrible name.
Right. And the thing about this lizard is Zootoca vivipara, as the name implies, it gives live birth. So it doesn't lay eggs, this little lizard. It gives live birth to offspring, except for 2 populations. One population in northern Spain and one population in northern Italy. And some researchers would argue, because those 2 populations lay eggs, now they lay very, very thin-shelled eggs, which don't do a lot of development because the baby's nearly already ready. Because again, it's a continuum. Laying eggs and giving live birth is also a continuum.
Mm-hmm.
And some researchers would say, well, maybe we should call these egg-laying populations a different species, but they're so similar otherwise that it really doesn't make sense to call them different species. So yeah, or as Brian Stephen points out, life, like nature, is probabilistic, right? It's all about probabilities. So what are your thoughts on this?
I think I would agree. Defining species is kind of a impossible game.
We agree with that.
Whether we're creationists or evolutionists.
Well, sure, but the problem isn't whether or not we're defining species. The problem is are things actually related? So whether you call, dog or your house dog and a wolf, a gray wolf, two different species or the same species, that doesn't really matter. What matters is, do they share a common ancestor? Does the word species actually reflect something in nature, right?
Right.
And so that's kind of what we're trying to settle with you. And it's perfectly fine to say, like, I don't know. That's perfectly fine. Dapper and I, there are lots of things we don't know.
Absolutely.
This just happens to be our point of interest.
I don't know why pelagic tunicates exist, but they do, much to my chagrin.
Yeah.
Yeah.
It's just one example.
The fact that there are blurry lines in defining families and species, I would say is interesting, but it's not enough to claim universal common descent. Just because we see similarities in body plans, that's a something we can extrapolate on and say that these are all derivatives of each other in some way.
So I have a question then, because when you, as you've said that you do look at certain groups of animals and say that you think that these are related, what are you basing that on, if not on similarities in the body structure? Because I'm assuming that you haven't run like genetic analyses, which is fine. I mean, that's a pretty technical thing that I wouldn't expect most people to do, but still, you have this intuitive sense that, like, say, all of Felidae or all of Canidae or all of Ursidae or all of Elephantidae are related by common descent. And I assume, and maybe I'm wrong here, that you're doing that not so much on the basis that they, this group ends in ide, but rather that they have this physical similarity. But if what you're doing is still using that to demonstrate or to convince yourself to your own satisfaction that those are related, that's also fundamentally what's going on with character-based cladistics, right? We're still taking a look and saying, okay, well, these similarities and differences and the patterns of them that indicate relatedness within Canidae also show relatedness between Caniformes or the wider Carnivora, or even wider Carnivoromorpha, or even wider, Ferae, or Ferungulata, or just Loraisatheria, as we just keep going up. And I'll stop now.
When you run those through, when you run like a character set, like a character matrix through a program to build a phylogeny, you're literally doing the exact same thing. Like, there is fundamentally no difference in including more taxa into a character. Like, so, by character matrix, I mean, you have like a table, and your table will say, like, does this taxon have wings? Does this taxon have forelimbs? Does this taxon have molars? Stuff like that. So you're listing the character traits. You're listing a whole bunch of character traits, and then you're taking different taxa, bears, seals, weasels, elephants, humans, and you're doing that, and you're checking yes or no for each of the boxes, or really you could say, like, 0 for no or 1 for yes. And so and then you take that and you have a program which then interprets that data and then puts it into a phylogeny. And so there's no difference doing that for, just bears, just ursids, for just seals, or for just cats, or just humans. You can put any number of organisms in there. Now, you want to do ones that will, give you a useful answer, a meaningful answer. Because if you put a bunch of random things in there, you're going to get a non-meaningful answer.
Yeah, if your cladistic analysis is just like humans, bristleworms, and like pine trees, you're probably not going to get a whole lot out of it.
Right. And so when you're doing that, you're doing the exact same thing. It's the same sort of analysis. And so you can do that for humans, for all primates, and you'll see humans nest within the great apes. Do that for all mammals. You'll see the humans nest among the primates, and then that primates are one set within the mammal tree, one of 4 super orders. There's again fundamentally no difference there. And so why should we stop? Why is it that we should only be able, or, well, let me rephrase that. Shouldn't we only be able to apply that sort of analysis to the kinds themselves and not between kinds if they were separately created? Like, why should we be able to pit multiple kinds against each other and get this phylogeny that incorporates all of them together? Why does that happen if they're not, if they didn't share common ancestry?
I think it's a good indication of universal biological language. You know, genetics is interconnected throughout all of life, and that's just something we share, and it's something that we have in common.
But that itself, it lends itself to the conclusion of common ancestry, because fundamentally we don't have to. We know because occasionally there are a few organisms, like some archaea, and also including the archaea that have become your mitochondria, have ever so slightly different translation mechanisms for their genetic code. And fundamentally, the genetic code assignments are essentially arbitrary. You could, in theory, have an entire group of life that uses an entirely different codon assignment set, or uses a different number of base pairs per codon that uses 4 instead of 3. Like, none of these are things that are, as far as we can tell, impossible. So the very fact that all life shares this fundamental aspect that is essentially arbitrary when you get down to the first principles, essentially, that in and of itself seems like indication of common ancestry.
Or, and also, yeah, piggybacking off that, that's also how we know that our mitochondria are bacteria, or were bacteria, because so when the first amino acid in our proteins is methionine. In both eukaryotes, so organisms with a cell nucleus, and archaea, which are this group of mostly extremophilic prokaryotes, they use methionine as the first amino acid in any sequence. However, bacteria use a different form of methionine called N-formal methionine. And also, your mitochondria, when it synthesizes its own little proteins, also starts its sequences with informal methionine. So that's, again, another way. It'd be super convenient, right, if it were not related to bacteria, but use the exact same codon assignment as bacteria. Right, that'd be really, really convenient. But a closer to home example than that would be there are things in our genetic code that are very low probability insertions, right? So our genome is in totality like what, 6.4 billion base pairs long.
I think it's 6.8, but I might be off.
Something like that. It's something like that.
We're in the right order of magnitude, so for this discussion, I think we're good.
It's about 6 billion base pairs. We'll say that. So there are regions in our genome where we have the exact same insertions as in other species, but not others, right? So, for instance, all rabbits have certain insertions in very specific places in their genome that only rabbits share, and nothing outside of rabbits. Sorry, yeah, like ERVs, like retroviruses, for instance. So these are viruses that insert themselves into our genome, and they don't pick a specific spot. There's lots of different places they could put it, and it's not totally random, but it's largely stochastic. It's mostly
good use of stochastic.
Oh, thank you. But only rabbits have these certain insertions. Now, we also have certain insertions within our genome that only chimps and bonobos also share, and then they're in the exact same place. And the same is true for gorillas, chimps, bonobos, and humans, and for
and
all
it's
great
fewer
apes.
of them.
Right, it's fewer,
and they also have their own unique ones that aren't shared.
And so, there's no reason for us to share these, right?
Well, apart from common ancestry.
Right. Apart from if common ancestry weren't true, it would seem very convenient for these to exist in the same places in all of our genomes if we didn't get them from a common ancestor. Do you see what I'm saying?
Yeah, I've heard that argument before. It's interesting. I would posit that it's a similar structure that is being attacked by the virus, and of course we share all kinds of
Okay,
viruses with
hold up though.
birds, and
I have to stop that for a second though, because the viruses are not targeting specific spots in the genome, which
Right.
is what you just said with
Right.
their similar structures. Retroviruses largely insert themselves, like Jackson Wheat said, at random into the genome. They do not have a preferred target area. So we cannot explain this away with structural similarities between, say, chimpanzee and human, chromosomes, because these retroviruses specifically do not target specific regions, and we can tell that because we can see current infections of retroviruses and where they insert in the modern genome, and two different people can be infected with the same retrovirus, and it will insert into completely different spots in their genomes, even though it's the same retrovirus. So we cannot explain this by simply saying, well, the retrovirus has a preferred target location, and that target location is similar because of structural similarities between chimpanzees and humans. That is not a viable explanation. We cannot go there. That has already been falsified. That is precluded by the data.
Right.
there's a video with what's his name, Dan
Dan Stone
Dr.
Cardinal.
Dr. Dan.
Yeah,
No, that
That's
he's
him.
actually a friend of ours.
Oh yeah.
Yeah. I know he was in that circuit. Him and Dr. Jackson,
Yeah,
Charles
we've
Jackson.
seen that video. Yeah.
Yeah. His explanation, I remember at the end was, I forget exactly what it was right now, but I go back and look at it. But that's what I would posit if you remember that video and that exchange.
Well, creationists try to give a couple different arguments, and none of them really work. Like we said, for the one, as also as Dr. Dan has pointed out, and also Dapper,
Thank you.
it's not totally random. Like it won't. I don't think it's going to insert in like what is it, heterochromatin or whatever. Or no, is it euchromatin? The one that
No,
doesn't
I believe it is heterochromatin, the one that's already like super methylated and tangled up.
Yeah, that's heterochromatin. Okay, yeah. I always get them confused. But it basically they probably won't insert there because there's like no way for them to get in there because it's not getting
Which is
transcribed or
why
anything.
you have heterochromatin in the first place.
Right, exactly. So it's largely random, though not entirely, not 100% random. But we do have retroviruses that insert people like on a daily insert into people and across the world on a daily basis. So we can look at different people and see these different insertions. Some people try to argue that like all of them are functional, which they really can't be because some of them are within genes, so they can't be like a different gene within a gene.
And further,
that wouldn't work.
They're inactivated due to mutations that stop them from functioning like viruses, but you can actually induce them to reoccur as viruses, which then can create a new infection. Now, you tend not to do this in humans unless it's cell cultures, but you can do this in other test animals like pigs or something, where you can actually reactivate these endogenous retroviruses to become viruses again that then start spreading again through the body. So we know they're viruses. They're not just functional designed elements of the original DNA of these animals, because why would they then be able to turn into viruses with a simple mutation?
Right, and most retroviral, the remains of retroviral infections are just what are called long terminal repeats. So it's just the long end sections of the virus, of the viral genes. Like the viral genes themselves are gone, like GAG, POL, EMV. They've been mutated out of existence, basically. But the terminal repeats, the ends, are still there, and they're just littered all throughout our genome. And we know they don't do anything. I mean, they're you can look at the transcription rate of these things. You can do you can take cell tissues, for instance, and do transcriptomic analyses. So you basically look at what genes are being turned on in the tissue. And that's in part how you figure out what genes are involved. So you're like, okay, this protein here is involved. You see that these genes are being turned on, or you find these transcripts. And so, okay, this transcript, we have to look for this gene. Aha, we found this gene. So these genes are being activated. So we can do that. We can see they're not getting turned on, at least not at any rate above like random noise, which happens. Suffice it to say, we also have these at many different levels. We have them within species, within populations, between genera, between families, between orders. There are certain ones that all mammals share in common, right?
Mhm.
And so again, this is kind of that sort of play on, like, where do we draw the line? And the point that Dapper and I are trying to make is that there just isn't one place. Try as you may, you can search the biological literature. You're never going to find a place that's like one for all, one size fits all. And that's kind of why I love biology. I think it's one of the reasons.
Yeah.
I don't know. What are your
I think
thoughts
it.
so far on this?
Yeah, I think it fundamentally comes down to if the fact that it's hard to define families and species shouldn't, and the blurry lines like this can't be evidence for any theory one way or the other. It's just kind of a part of what we see life is. It's just,
You say the fact that we can't define these categories taxonomically means that that's not evidence for either evolution or creationism?
No, the fact that we can't find a one catch-all definition that's tight and robust and
Okay.
that's reason to say, oh well, it just must mean that everything is related, and because they share these things, and that
Well,
the lines are blurred. That's just
well,
not
again,
good.
That's not why I'm saying that these organisms share common ancestry. I'm saying now, I agree that, I taxonomic categories are going to be arbitrary anyways, because the guy who essentially founded the concept, Carolus Linnaeus, believed that species were fixed, by God in the beginning and can never be changed.
Oh yeah,
So,
and far more specifically than you're thinking, Monk Man. He's thinking like the gray wolf has always been the gray wolf. It shares no common ancestry with the jackal or the coyote or foxes. Like God created gray wolves, and he separately created jackals, and he separately created coyotes. Was his idea,
right? And right, yeah. So what the reason that we and biologists, 99% of biologists would say that
come now, 99.9. Give it another order of magnitude to be more accurate.
Yeah,
it's far less than 1% of biologists disagree with common ancestry.
You're right. I was just shorthanding it. But the reason that they agree is it's these multiple tiers of reasons for thinking that things are related, right? So the reasons that you think all bears are related, by whatever category, is also the reason. So that is shared with seals and weasels and dogs, right? There are morphological characters that unite all of these, and genetic characters, and developmental characters, and fossil characters. All of these characteristics together, all these independent lines of evidence that don't necessarily have to correlate with each other, right? There's no reason necessarily that genes have to correlate between bears and dogs or bears and weasels. There's no reason whatsoever if they're created separately.
I have a good instance of a place where genes don't correlate despite making very identical results, right?
Shoot.
So both echolocating bats and echolocating cetaceans, that is primarily dolphins, make a protein called prestin. And from a protein sequence, prestin in both of these animals is identical. But when you look at the neutral variation in the genetics that makes the protein, there's no correlation that's any more than just sort of random chance, which means that we have an example of what it would look like if we could say that separate ancestry is likely, because in terms of just making prestin, that's for echolocation, bats and cetaceans, in fact, do have separate ancestry for this one particular protein. So they settled on the same protein, but with different genetic backing. And there's no reason, if separate ancestry were true, that just wouldn't be the case for proteins generally across in different kinds. So, for instance, there's no particular reason why, say, I don't know, Jackson, what's a good protein?
A good protein? I don't know, cytochrome C.
There you go. There's no particular reason why cytochrome C, in terms of neutral variation between, say, a sea lion and, say, a weasel, would share any more in common than is strictly necessary just to get you a functional cytochrome C, unless there's common ancestry. But whenever we actually do run statistical phylogenetics or cladistics on these things, we find
Using
that in fact,
cytochrome C.
right, cytochrome C is actually a common way to do this. We actually do find this commonality that there's no reason to expect other than common ancestry, unless you want to go ahead and say that the creator just designed things to look like common ancestry as like a little trick, like haha, fooled you. Which, if you want to go that far, now you're allowing an omnipotent trickster deity into your epistemology, which basically means you just can't know anything.
Well, there's counterexamples to homology, convergence, and
I would love
you know,
to hear one.
How do you mean? Can you give it? What do you mean by that?
let's see. Gosh. AIG has a few of them. See, is
Remember
it?
any?
Yeah, there's I want to say a squid, or yeah, a squid that shares some type of deep sea amphibian that has similar eyes to humans. And yet we're
So the eyes they look sort of similar, but fundamentally cephalopod eyes and vertebrate eyes are actually constructed backwards.
Yeah,
They do
they're
look
actually
similar in a lot of ways, but they are backwards of each
Yeah,
other.
they also don't share strong genetic homologies, which is what
Right.
we would expect, because
It's totally different. Or aside from like the very core genes involved, like PAC6, it's pretty much all different.
yeah. So this actually fits very well with the expected like genetic and also character based phylogenies, where superficially these eyes function similarly because
Right.
the physics of light is the physics of light. It's not going to change based on your ancestry. So to solve the problem of how do you see, there's only so many solutions. But when you actually dig into both the genetics and the actual structure of the eyes, they're actually fundamentally different in very important ways that are exactly what you would expect from common ancestry.
and that was the other thing, is that there was a subspecies of that squid, we'll call it, that had different eye structure, so it wasn't the same
So
lens, and so that's a
What do you
That's-
mean by not the
So-
same lens? What is that?
Well, for one thing, eye structures actually evolve. Eyes have evolved very many times and are actually relatively easy to evolve in different groups and even change. So, like, there are fish that have eyes that hang off of their eyes. There's this one guy called Bathylicnops exilus. So he has his normal eyes, which point forward, but there's basically a second set that hangs off the bottom of the eyes that point downwards. And developmentally, they appear after. Now, basically, what it comes down to is they're really just sort of a piece of the retina that has sort of split and has its own separate cornea, which is not cornea, separate. No, you're right. A separate lens, at the very least, has a separate lens, which is also not developmentally related to the lens, to the normal lens. But since Bathylicnops exilus is in a group of fish that don't have this structure, I think by your criterion, that would be within the range of like possible development, right? Like that would have occurred after their creation, right?
Yeah, I'm trying to find this article.
Oh, and I'll put it in the side chat what the name.
Oh yeah,
is.
yeah. If you put that in the internal chat called
What
private
not?
chat there, that would be very interesting to check on.
Okay.
But
Yeah.
I mean, so are you
Eye
saying
structures change a lot. There are like worms, or sorry, flatworms with compound eyes. There are flatworms with like concave eyes. Eyes change very rapidly in different groups of organisms. They're very malleable.
yeah, and fundamentally, I mean, unless you have a counterexample, they seem to always fit very well with our various twin nested hierarchies between both morphology and genetics, because that's one of the important things that we've been actually talking about, is the twin nested hierarchy. Similarity in morphology is mirrored in a way it doesn't have to be by strict necessity in genetics, because one of the things, do you know what synonymous codons are?
I don't.
Okay, so do you know what a codon is?
it's familiar. I don't know off the top of my head.
All right. Well, I'll give like the 50 miles level of like overview here. So a codon is a group of 3 nucleotides, which is, you know, the A, C, T, and G in your DNA. Every 3 of those forms what's called a codon, and each codon has an assignment that's either a start or stop codon, or it codes for a particular amino acid. Now, most groupings of these, like for instance, if you pick a particular amino acid, it has more than one codon that codes for it, because there are more codon options than there are actual amino acids that are used in living organisms. So most codons share their amino acid assignment with another codon, which means if you have one codon versus the other that are synonymous, that's what those are called, codons that have the same amino acid, the protein that results is completely the same, right? So which one you have doesn't matter in terms of the protein. However, if you have common ancestry, you would expect that the codons used to make a particular protein in you versus someone else that you share common ancestry with would be usually the same codon if you're getting the same protein out of it. But if you don't share common ancestry, but you're still making the same protein, there's no reason to think that you would have similar codons in terms of which ones are synonymous. So for instance, you might have, for one amino acid, you might have this particular codon, and then someone else who's not related to you at all, like say if you're not related at all to a bat, but you're making the same protein, then you would expect that generally speaking, the codons that they're using to make the same protein as you would generally be uncorrelated. And yet, when you actually look at neutral variation like this in synonymous codons, you actually still reconstruct the same phylogeny that you usually get with both character-based as well as just other types of genetic ancestry. And so there's no particular reason to think that this would be the case apart from common ancestry. And so it's all of these different things, right? It's not that it's one thing. It's not just ERVs or just the fossil record or just morphology. It's the fact that all of these converge on the same answer so reliably. And that is not what we would expect from separate ancestry. In fact, it's hard to imagine a stronger refutation of separate ancestry than all of these things coming together at the same time, because they don't have to, right? There's nothing about the biology of humans that says that they have to have the same ERV insertions or the same synonymous codons as chimpanzees in most cases. And yet they do. And there's no reason to say that they should have similar to gorillas, but less similarity than with chimpanzees, except for common ancestry. And yet they do. And that goes through all of life. You can take that all the way to like bacteria. You share genes with bacteria that are more similar than we would expect, except for common ancestry with those bacteria. Why is that if not for common ancestry?
Yeah, that's pretty deep for sure. Probably a little bit beyond my pay grade terminology wise. But yeah, I would say, it's genetics is it's a shared language. It's a universal system with
It doesn't have to.
individual. What do you mean doesn't have to be?
So
Go ahead, Jack.
If the kinds were created separately, then an omnipotent God could create every single kind with its own genetic code, totally unrelated to anything else. It wouldn't be hard. It's definitely doable for an omnipotent God. But
Go ahead.
That's the opposite of what we see. Not only do all these organisms share genes and DNA, not just DNA, but like RNA, and we all share ribosomes and proteins. On a fundamental level, all life shares a series of like macromolecules, right? There, again, no reason for that to be the case under separate ancestry. It would be very, very easy for an omnipotent creator to endow every single kind with separate genetic material, right?
Which would itself be extremely strong evidence of common ancestry, of separate ancestry.
Right, exactly.
Like, it would be hard to refute if the omnipotent creator had just decided, hey, I'm going to give fundamentally different genetic like codon assignments to every organism. And you can say, oh, well, they wouldn't be able to eat each other. Except, yes, they would, because you can still break down the proteins and the lipids and whatever that they make. That's not affected by this.
it's funny that you bring that up because there's actually a I forget the name of the organism, but I want to say it's in the ocean that doesn't share any biological information with any other animal on the planet.
I don't
am
believe you.
skeptic. I don't buy that.
It was brought up to.
I'm going to make it a stronger statement. It's not that I don't believe you. I believe you are incorrect. That is a stronger statement. Unless you can name it and show a reference for that, I'm going to say that that is incorrect.
Oh, sure. Yeah, no problem. It was brought up to Richard Dawkins by
that shares no common ancestry with anything.
It shares no.
You said this is an animal?
Shares no. It's not an animal. Hold on. It's in my
Okay.
YouTube playlist. It doesn't share. Yeah, I'll just look at it. I'm not
the thing is,
in the meantime, I'll distract you all with my juggling.
If this were true, I would expect it to be the most famous organism in the history of science, because that is extremely unexpected. And what I'm fully expecting is that you are going to produce an organism. Either you're going to not produce it because you
not the octopus. Please don't be the octopus.
Oh my goodness, please don't be the octopus. Or what you're going to do is produce an organism which actually fits very neatly into nested hierarchy, despite weird claims about it coming from like panspermia enthusiasts. Because I think I know what we're going for here, and I think it's the cephalopod panspermia.
I hope not. Oh, I hope not. Please.
I found the video.
All right, what animal or organism are they referencing?
Yeah, I'm gonna see. Hold on, if you can hear it.
DM Wing says, "I bet it's a mollusk." Yeah, it probably is.
I can't hear that.
Do you know from your work what the minimum? Do we yet know what the minimal configuration is to make life?
You mean the minimal gene set?
The minimal gene set.
Can you hear it? Can you hear that?
It's a little echoey, but I can kind of hear. We're talking about minimal gene sets, which doesn't sound like.
Hold on, he's
talking about.
He's getting to it.
Okay.
Whittling down on it, but there won't be a minimal gene set. There'll be multiple ones because I'm not so sanguine as some of my colleagues here that there's only one life form on this planet. We have a lot of different types of metabolism, different organisms. I wouldn't call you the same life form as the one we have that lives in pH 12 base that would dissolve your skin if we dropped you.
Oh, I've got the same genetic code. We'll have a common ancestor.
Well, you don't have the same genetic code. In fact, the mycoplasma uses a different genetic code and would not work in your cells. So there are a lot of variations on the
But you're not saying it belongs to a different tree of life.
Well, I think the tree of life is an artifact of some early scientific studies that aren't really.
So that was mycoplasm. Did you hear that?
Oh, so mycoplasma is a bacterium. It is very much nested among the other bacteria.
I will say I am very happy that the answer that we were getting is not just cephalopod.
Yes.
Because that's what I was expecting, which is one of the cringier claims that I've ever encountered in biology. So
mycoplasma is an interesting little bacterium because, for one thing, it does a version of multicellularity, which is kind of cool, which is why it was actually mistaken for a fungus, which is why it's called myco, which means it relates to fungi, mycoplasma. But no, mycoplasma is just a normal bacterium. It fits very well within. Now, what he's saying is the tree of life is an artifact, and for prokaryotes, that's probably true. It'd be very interesting to see like a strict tree of life for prokaryotes because they share genes so frequently. But no, mycoplasma is not like totally weird or out there. So no.
So I do want to mention that there was mention of it using a different genetic code, and what that means is, do you remember what I was talking about codon assignments and synonymous codons and whatnot? That was a question for you, Monk Man. Did we lose him?
Monk Man?
Uh-oh. Monk Man?
Whoopsie.
Oh,
I accidentally closed it.
welcome back. So what I was saying is, do you remember when the speaker? Which, by the way, who is the speaker there?
Dr. Craig Vetter, is that what it is?
Venter? Yeah. Okay.
Okay.
Yeah.
Okay. So do you remember when he said that this bacterium was using a different genetic code?
Uh-huh.
I think he was just getting to it. I
Yeah.
can.
So I want to go into that because what he's talking about there is, do you remember when I was mentioning things about like synonymous codons and whatnot?
Mhm.
So there's this thing called the consensus code, which is the most common codon assignment that occurs in living organisms. But we also know that some organisms use a variation on that, and those are non-consensus or non-standard genetic codes. But the thing is that they're usually only varying from the consensus code by 1 or 2 different codon assignments, which means that, like, with you, a particular codon might code for this particular amino acid, but with some organism, it codes for a different one. And we rarely get beyond 1 or 2 different changes in codon assignments, which itself, the fact that there's such a strong consensus within the genetic code, such that there is a consensus code, and that even when we get variations from it, it's usually in very small amounts, is on its own another reason to suspect that common ancestry is true. Because again, codon assignments don't have to be what they are. There doesn't have to be a consensus. Every kind could have its own codon assignment that's completely at odds with the others.
Right.
And there's no fundamental chemical reason why that couldn't work. And yet that's not what we see. Instead, we see fairly restricted deviations from the consensus code, and usually even then, very small deviations. So even that on its own is hard to explain with a separate ancestry model, whereas it fits in with the predictions of common ancestry, which is to say that the consensus code should be by far the most common, and when we see deviations, they should be both taxonomically restricted and relatively minor in terms of like variation in codon assignment. So even that, which also, by the way, it did not conclude that this organism shared no commonality between other organisms. That is not what was said in that video.
Yep.
But even again, we're still seeing things that are very well aligned with the expectations of common ancestry.
As Nestle put in the side chat here, basically the stop codon and the tryptophan codon swapped. So that's basically it.
Okay.
Which
Yeah,
is in line with what Dapper said.
So, yeah, we're getting 1 or 2 different assignments.
Yeah. I definitely misdefined what was going on there. but it's a
happens.
The point was it's kind of a deviation from.
Well, sure, but
Yeah.
there are codons in your mitochondria that are like that. There are some animals that have, like, again, 1 or 2. It's a few. It's a handful. It's not that many.
And they remain taxonomically restricted, right? Like it's not like
they're not randomly scattered.
Right. It's not like every family of animals has a different genetic code assignment, right? And even within mitochondria, the mitochondria of eukaryotes, as far as I know, shares the same slightly variant codon assignment, which itself is a taxonomic restriction, unless I'm wrong about that, Jackson.
And Sai points out, because Sai is a biochemist, that usually these variations are only for stop or tryptophan, not for the more important amino acids.
Right, which is another thing. Like the ones where evolution would have the least problem changing things because it would cause the least problems are the ones that tend to be swapped out. So across the board, we're getting taxonomically restricted minor variations in the universal or consensus genetic code, which is not a prediction of separate ancestry. It's a prediction of common ancestry. Now, you can accommodate it in separate ancestry, but that's actually part of the problem. Because if you accept separate ancestry starting from an omnipotent creator, I don't know what data would disconfirm that. But the fact that it's unfalsifiable on its own is actually a problem because it removes it from the realm of science and puts it into pseudoscience. So what we can do, though, is we can say, well, since there's no reason under separate ancestry to expect there to just even exist a consensus genetic code, or for organisms to have a similarity in neutral variation that corresponds to their morphological differences in like a phylogeny, and because there's no particular reason to expect transitional fossils between known extant groups or even known extinct groups, yet we do find them, because all of these things are true, and there's no reason to expect them under common ancestry, even if common ancestry can accommodate them, that in itself indicates that while we can't technically falsify separate ancestry, that is not the most parsimonious or the most likely inference to make about biology.
I'll also tell you, as someone who is going through a lot of paleontology papers right now,
Oh yeah.
when you are looking at two lineages and you are finding where they meet, like you trace them back to their common ancestor, and like in the fossil record, the fossils you find on either side of that branch point are very, very similar to each other, and that's true
Virtually
all
identical.
across the tree of life. Yes.
My favorite example there is that it takes expertise with years of study to figure out the difference between rhinos and horses once you get back to the earliest members of those groups. You have to examine their teeth like with a microscope to tell the difference between the earliest rhinos and the earliest horses. It's absurd how identical those animals are.
Right, and the same
Really?
is true for
Oh, yeah.
the same is true for like the first placental mammals and the first marsupial mammals. I did a video recently on called the placental mole's tail, or the marsupial mole's tail. Sorry,
There you go.
and in that I talk about the differences between the first placental and the first, the earliest ancestors of marsupials, and the difference comes down to, again, their teeth. It's literally just their teeth. Everything else is more or less identical.
Tiny, almost microscopic differences in dentition is often what differentiates the earliest groups of different mammals from each other.
Yeah.
And like, if you were to look at the first rhino and the first horse that we have in the fossil record, they're about as different as like a wolf and a jackal. Like, if you looked at these animals, you could confuse them for each other.
Yeah.
Because
Yeah.
they're almost completely the same, which is a prediction of common ancestry, but there's no reason to think it's true under separate ancestry. Why should the earliest examples of fossil horses be essentially identical to the earliest example of fossil rhinos, to the point that you have to have essentially a PhD in the difference between horses and rhinos to tell them apart? Like, that's the level of expertise we're talking about. People spend their careers figuring out whether a tooth is from a horse or a rhino because it's not easy.
And I've had a guy on my channel. His name is Donald Prothero, who does that. He does that for a living. That's his whole job. Yeah.
I mean, it's amazing how identical these things are, and you get it all over the place, right? Like the earliest Sirenian, which is the group that includes like the dugong and the manatee, and the earliest Proboscideans, virtually identical. You look at their fossils, and it's just like, that's just the same animal. Like, what are you talking about? Like, the difference between the earliest Proboscidean and the earliest Sirenian is like, the earliest Sirenian is a bit shorter and longer, and they have slightly different teeth.
And eats freshwater plants as opposed, the earliest Proboscidean eats, freshwater plants, whereas the earliest Sirenians ate like saltwater plants.
Right. Like, it's at least as similar as like two different species of goat, right? Like, it's essentially identical. And why is that, right? It's under common ancestry. It's not just explainable, it's expected, because the further back in time you go, the more closely related different groups are. Like, the actual more close related, like if you did a genetic sequence of them, they would come back as being closely related. They were very similar,
and in
compared
fact,
to their modern descendants.
You can't. That is, again, another prediction is that the further you go back in time, the closer sequences will get to each other, right? And so you can also do that sort of test genetically using extant organisms, and it still bears the same results. Because there's a paper that did that. It was like Beyond Reasonable Doubt, I think was the title of it, something like that. But yeah, you can do this with these different methods, and you keep getting the same answer. So we'll only go about another 18 minutes. I don't want to hold you for too long. So at this point, what are your thoughts, Monk Man?
I had a couple questions, if I could.
Yeah,
put you on the hot seat.
I love questions.
Let's see. We left off talking about the notochord on, can't remember the name of those early fossils, but
like Haiku or Haiku.
Yeah, one of those. I was curious, what would you say happened to the notochord, and then insofar as becoming the the backbone, or in how did that transformation go about? If you could kind of model it.
Well, I mean, so if you look at just your own ontological development, right? Your skeleton starts out as a cartilage thing that then calcium is, well,
It
you know,
ossifies.
hydroxyapatite, which is primarily calcium, is deposited around those cartilage structures. And we know, because we can watch both genetic diseases as well as extant animals with different levels of ossification on their cartilage skeletons, that is the kind of thing that mutations can affect. And so if you have a notochord and you have a mutation that causes calcification of parts of the notochord, that is the beginning of a bony spine. And we know that happens, right? There are living animals with different levels of calcification of their skeletons. In fact, that varies within humans. Even humans have variation in how much of their cartilaginous skeleton ends up ossifying. Now, in many cases, it ends up being pathological if you have too much calcification. But still, we know fundamentally that mutations can cause differences in calcification of cartilaginous structures. So, like, if that's the question, then the answer is it ossified.
I mean, you can look at, if you compare like sturgeons and gars with like the quote bony fish, like, you know, goldfish, trout, cichlids, you can see that there's differences in the degree of ossification that occurs.
Is that a turning on of a gene that's already existed? I hadn't heard of that mutation being able to do that.
I mean, I don't know all of the genetics surrounding ossification of cartilaginous skeletons.
What do you mean? Is there just a mutation that turned on like a gene that, I'm not sure what you mean. Like you could do deposit, so like lots of organisms have genes for biochemical pathways that deposit calcium. It's just a matter of, and that way predates chordates. It's just a matter of where are you depositing it specifically is more of that point. It's, you've already got calcium in you. It's just where are you putting it?
Right. So that that placement is a pretty significant structure in the genome, gene placement. And that's not something that I know of. You can just move around, and that just willy-nilly you can add calcification over here, not here. It seems natural selection is, you know, what's already in the genome.
Well, so look at snails, for instance, right? Snails have shells that are made of largely calcium carbonate, right? The degree of change of like where you're putting it varies enormously among snails. Some have these like inline coiled shells. Some have like these spiral shells. Some have very thin shells. Some have very large shells. That's, you know, very much the result of mutations. And in fact, within a single species, the direction of coiling of the shell can be inherited through Mendelian patterns, right? So your sperm you get, you know, a sperm cell that tells you either coil the shell left or right. That can vary within a species. So I mean, yeah, it's very much a product of the mutations and stuff like that.
Yeah. I'm a little confused. Is your contention that genetics does not control calcium deposition in living organisms? Because if it does, then I don't see why you would say that mutations can't change that, because fundamentally, if genetics controls the thing, then mutations to those genes can change the thing it controls.
It does for existing features. If something, you know, like the notochord in lamprey larvae exists when they're young, and then when they grow, it whatever dissipates.
Sure.
And the backbone replaces it. It's basically like a support structure, kind of in the meantime. But that structure was already there, right?
So a lot of times I think I see where you're going with this. Let me see if I can try. A lot of times for structures, they're not new. They're not made of a bunch of new genes. A lot of times in biology, structures are made of genes that already existed, but which are being what is called co-opted or accepted for new purposes. Okay.
Mhm.
So for instance, the genes that control arm, leg, hand, and foot development are in fish, controlling fin development. It's the same, it's what homologs of the same genes. So like Sonic Hedgehog and all these sorts of things. Those are the same genes in fish which are controlling fin development, but which have been modified through mutations, which are now doing arms and legs. And you can compare it of like a goldfish. You can compare the Sonic Hedgehog of a goldfish versus that of a lungfish, and you can see that the lungfish Sonic Hedgehog is much more similar to ours than it is to the goldfish, right?
Right, but we're not talking about modification. We're talking about
That's
brand
most of
new.
evolution. It's very rarely in evolution do you get wholesale new things. So a lot
Well,
of times,
I agree with that. That it's that natural selection is just modification, but I
natural
digress.
selection is a selecting of the modifications that arise through mutations and recombination. So, like, you get duplication, for instance. So, like the hemoglobin. So the chains in hemoglobin are duplicates. They're like lots and lots and lots of duplicates, and they have been modified to have different functions. But if you do a phylogeny of the genes, you can see that they all trace back to a common ancestor that got duplicated originally and then got duplicated many more times. And you can do that for lots of different genes.
Right, and those are modifications. Those are existing sequences in the genome that
Right.
the ancestor had, and that was then selected again.
Right.
You know, maybe in a
but
different.
that's fundamentally how evolution is thought to work, right? Like,
But you're suggesting that a new structure, the backbone, wasn't there at one point, and then all of a sudden it was.
But the
Well,
thing
not
is,
That's not a
all.
modification.
the backbone is itself a modified notochord, right? You can
Right.
call it a new structure all you want, but fundamentally, everything that you can point to is a modified other thing that came before, all the way back to the earliest life. It's sort of like saying that this is some kind of problem, right? That these are just changes in previously existing genes. It's sort of like if I objected to Christianity by saying, yeah, but Jesus was supposed to have lived in, the Roman province of Judea, and that totally existed, so Jesus didn't exist. It's like, no, that the existence of Judea is a prerequisite for Christianity, but it's not a counter. Similarly, the fact that evolution says that things that occur that are you might consider quote unquote novel are modifications of previously existing structures is just the way evolution is supposed to work. That's how it happens. You can't say that that's a problem for evolution when that's just the way it works.
Right. Hedgehog itself is a combination of different genes. Like, I think it's hedge and hog, which were different genes originally, which then got, for whatever reason, fused together, and then underwent duplications and modifications in different lineages to fulfill different functions. Now, yes, now you can say, okay, well, that means that, like, fins and human limbs are the same, or, they aren't, neither of them are novel because they're both derivations on Sonic Hedgehog. But, like, I think you kind of lose the plot at that point because they are different. Obviously, they're being used for different ecological functions, for in different life histories and all that sort of stuff. So I don't think it would make any sense to say like fins and limbs aren't new or aren't fundamentally different from each other. You know what or they aren't different from each other. Sure, fundamentally they aren't. But do you see what I mean?
Right, but in this case, the example I gave, the notochord is existing alongside of the backbone.
Well, yes, the
So
backbone is a structure built atop the notochord.
It's built atop and from the notochord, which is another important thing, right? Because remember how we said
Is
there
it
are
really
different
from
levels-
the notochord? I'm
Yeah,
not.
because remember how we said there are different levels of ossification? The notochord, because there's a selective pressure, right, to keep a cartilaginous core to a spine for reasons of like flexibility as well as preventing bone from rubbing against bone, things like this, right? So there's a good reason not to ossify the entire notochord, but if you watch embryological development of a spinal cord, it starts with a notochord around which is deposited hydroxyapatite, biohydroxyapatite, which is just the calcium that's forming around that to form your bones. That's what's physically happening. You can watch it happen. You can, if you had a good microscope and some kind of animal embryo that you could watch, probably a fish, because they have relatively easily visible embryos with their clear eggs and whatnot. You could yourself in real time watch a notochord have calcium deposited onto it to become a spine. Like, this isn't some kind of like hypothetical ivory tower thing. You can just watch it if you wanted to. Now, granted, you would have to get yourself a really good microscope, and those aren't super cheap, but like,
And you got to watch for a long time.
right, you're gonna have to keep watching it. I mean, like, this is not like
you could do a video, I guess.
Right, this isn't like an afternoon project that doesn't cost any money, but still, it's fundamentally the case that you can just watch this happen. If that were really important to you, to find out what happens when a notochord becomes a spine, you could just do it. It happens daily, all over the world. Notochords become spines.
Peter makes, or sorry, Nestling makes a good point. Peter just put it here in the side chat. The bones, the three little, or well, two of the three bones that make up your inner ear, the malleus and incus, were actually themselves jaw bones. They were bones that were part of the jaw, the lower jaw, and they are still in reptiles and birds, but they moved upwards in mammalian ancestors to become part of the inner ear. And that's why when you look at the platypus, for instance, its ears are much lower to its jaw than like placental mammals are.
Mm-hmm.
That's part of the reason.
Also, embryologically, that's another example.
Yes.
Those bones start it physically located in the lower jaw of mammals, and then migrate into the ear during embryological development, which is also mirrored in the actual adult states of various synapsids in the fossil record, where we can see these jaw bones, which I don't know if you know, but like typically jaw bones are part of hearing in non-mammal tetrapods. So like frogs and salamanders and lizards and whatnot, they hear at least partially through their jaws. And so you can watch in the adults of these synapsids, these bones shrinking and moving further and further to just the very back of the jaw, and then moving up into the ear in the adults, mirroring the embryological development, which again is all predictions from common ancestry. And in fact, if you look not that far in the past, you can find creationists saying, well, we'll never see a transition between the reptile condition and the mammal condition in terms of like the ear bones or the jaw bones. That's an unbridgeable gap. And yet, there it is, both embryologically and in terms of
Probanignathus, Diarthrognathus. Yeah,
right,
there's
in terms
lots of fossils.
In terms of both fossil intermediates and embryology, we see that literally happening. And so this is the frustrating thing for me, right? It feels like no matter how many confirmed predictions of common ancestry occur, people who are, for reasons other than evidentiary reasons, committed to separate ancestry will never care. And I'm not saying that that's you, Monk Man, but it does feel like it's the case for virtually all of them, that they have this conclusion that is not based on evidence. It's just based on some prior conclusion that they've decided for other philosophical or theological reasons. And no matter the evidence, they will say, and then they'll make up a post hoc rationalization. But the fact remains that common ancestry makes predictions which don't have to be true. There's nothing that fundamentally precludes other options from being true. Common ancestry is eminently falsifiable. And instead of being falsified, its predictions come true time and time and time again. And the response that is generated from people who reject it is just, meh, but I don't really care. And then they come up with a post hoc rationalization as to why the creator did it that way, or why it doesn't really count as a prediction, even though they didn't predict it, but evolutionary biology did. And so I want to make clear, I'm not trying to accuse you of being in this camp, but what I am saying is that it gets tiring, is basically what I'm saying, where it's just like, it fundamentally comes down to the same thing.
Yeah, I agree.
It comes down to the same thing as the flat earth. How many pictures of the Earth curve do you need? How many examples of measuring the height of buildings and mountains do you need before you believe the Earth is curved? And the answer is there's no amount, because fundamentally, what creationists and flat earthers have in common is they don't, from my observations, they tend not to care about the evidence. Instead, they have some pre-conclusion that they are dedicated to for non-evidentiary reasons.
But let me try a simpler question.
Go for
Sure.
it.
What do you define a mutation as? In a quick one or two
Definitionally,
sentences, kind of.
A mutation is any change in a nucleotide sequence.
Yeah, I would agree with that.
Any change, huh?
That's just literally what the definition of mutation
Deletions,
is.
duplications, insertions, substitutions, all of
Any
these are all mutations.
Yeah, in a nucleotide sequence is definitionally a mutation.
Because
Doesn't matter if it does
Wikipedia,
anything or not.
because Wikipedia, and I know we quoted from there earlier,
Sure.
so it says it's a copying error. So
Okay, sure, right. So mutations happen when like you have DNA polymerase and it's making a new copy, because you start with a template strand and then it attempts to copy that, and so in failing to do so, you have sequence changes, and that is the mutation.
Yeah.
So yes, I agree. It is a copying error. It's just saying when that's happening. That's basically all it's adding.
Now, I want to point out that the word error has negative connotations, which I don't want to be importing into genetics,
Right?
Because genetics doesn't claim that there's an ideal genome that exists. So when you say error, we're talking about the quote unquote
the one that
of transcriptase, or of this transcriptase, right, is to make a perfect copy. But when it doesn't, mutations occur. But that's not an error in the sense of like there was the perfect version and now we have the degraded version. It's just an error in the sense that it has failed at the fundamental job of making perfect copy.
Right.
Right.
Because
And
most mutations don't do anything. They do nothing at all.
Oh yeah. Yeah, they're mostly neutral.
Right.
Overwhelmingly.
Right.
Which is why I'm baffled as to how they end up creating these vast, complex structures from any amount of time at all.
So there's actually a really interesting burgeoning field within genetics called constructive neutral theory. So you have neutral theory, and that's basically the idea that, well, okay, in its pure form, neutral theory basically said that most mutation fixations were by genetic drift, which is debatable. But constructive neutral evolution or theory, whatever, basically says that you have these neutral mutations occurring at, well, most mutations are neutral, and you can get to a point where a mutation happens which causes a sequence to become selectable, right? So you're occurring a bunch of mutations and they don't do anything, and then maybe you have a promoter which moves to a new area, which does happen a lot, and this promoter in this new region is now causing a sequence of nucleotides to be transcribed to make an amino acid sequence. That happens. Yeah, you're right, Peter. We're hitting 2 hours. Okay, we'll try to wrap this up shortly. But so constructive neutral evolution, you can compare sequences within different organisms. Like, so one really good example, someone mentioned it in the live chat earlier, is that, so for instance, there are these ice fish that have these antifreeze proteins. So these fish live in Antarctic waters, and they don't develop ice crystals in their bloodstream because of a protein. And this protein is originally derived from 3 amino acids that came from a pancreatic protein, which was basically function in degrading trypsinogen. So it degraded. So originally, this pancreatic enzyme degraded a protein, but 3 amino acids were cleaved from it and placed elsewhere in the genome, and then they were duplicated over and over and over and over. Now, for the first, they got duplicated like 40 times, something like that. But it's a very tiny sequence that got duplicated over and over again. Now, for the first bunch of duplications, it didn't really do anything at all. But after this sequence got duplicated however many times, it conferred the ability to basically prevent ice crystal formation because the sequence is threonine, alanine, alanine, which was originally taken from the pancreatic enzyme, and that prevents ice crystal formation from happening in the blood. At least it lowers the temperature needed to completely freeze the bloodstream. And so then you can just keep duplicating that segment again and again and again. And when you do that, you increase or you make it more and more difficult for ice crystals to form in the bloodstream. So it took several duplications of this before it actually became useful. And then there was selection to preserve it. And then it really took off. Does that make sense?
Yeah. And would you categorize that that effect as what's going on behind the sequence itself? Was that a turning off of a gene?
No, it didn't turn anything off. It duplicated and just sort of moved the little duplicate over. That's what happened.
And that
So nothing was turned off.
Well, something had to occur to change the blood flow. That's essentially
No.
what?
No, the blood flow is still the same. It's still flowing in the same fashion. This also, based on molecular clocks, appears to have coincided with when Antarctica was getting colder. So there became a selection pressure to preserve genes that would delay or stop ice crystal formation as the ambient ocean was getting colder. That or just swim somewhere else.
Right.
So
yeah, polar bears have the same kind of mutations for fat storage and the color of their fur. Those are both
antifreeze proteins, though.
Not for antifreeze proteins, yeah, specifically, but they have they have their own adaptations for
right. Which on its own, the fact that there are different cold weather adaptations that are very different across widely dispersed taxa is on its own curious, because if you're going to go with, same designer, same design, why don't polar bears just rely on antifreeze proteins? Why do they instead rely on things like fat reserves and high metabolic rates? That on its own is curious.
It's a big genome. Yeah.
Yeah. So where were you going with the with your question about it?
Sorry to be disruptive.
So my point here is that no, I don't see any beneficial mutations, and in fact,
beneficial.
I mean,
so beneficial in the sense of new function. And so that's really where this distinction comes down to.
That is a new function.
Yeah, antifreeze is
the
a new
original.
function.
Yeah, the antifreeze didn't exist originally. It was an enzyme for breaking down proteins originally. It just so happens that the amino acid backbone of this, that 3 amino acid sequence, just so happened to also be able to function in ice crystal. They're preventing ice crystal formation. Yeah.
Yeah, which with also only after mutations, right? It didn't start out being able to do that. So
I'm not sure. I can't fully understand that this mutation.
I'll break it down as simply as I can. Right? Fish starts out being able to break down some proteins with its own protein. Fish has duplication event. That means that now it has 2 copies of the protein that breaks down other proteins. At this point, the fish has no ability to inhibit ice crystal formation in its cells, right? None.
Ice crystal form. What is ice crystal formation?
It's the freezing of water.
So So it's
water gets cold enough, it turns into a crystal.
In the bloodstream, this happens.
Well, anyway,
it can, yeah, if you freeze,
right.
yeah.
Like if the temperature gets low enough and nothing stops it, water will turn into jagged crystals because that's just physics.
Right? Okay. And they have this function already, or they do not?
No, they do not. They have a protein that breaks down other proteins in their blood. That's all they have. Then they duplicate the gene that makes that protein. So now they have 2 copies that just make a protein that breaks down other proteins. They don't have any ability to inhibit ice crystal formation beyond just having somewhat salty blood, which technically inhibits ice crystal formation, but that does that in anything that just has blood with any salt in it.
Right.
So, like, you have that ability too, right? If your blood gets to 0 Celsius, you're not going to quite have it form crystals yet. You have to get lower. But that's just true for animals generally. Okay, so now one copy of this gene mutates. And suddenly, instead of breaking down other proteins in the blood, it inhibits ice crystal formation, which was not a thing that it could do before. And it also still has the original copy, or one of the copies that still makes the protein that breaks down other proteins in the blood. So now it still has the original function. Plus it has another function with a new protein that's related to the old protein, but is still itself not the same protein, that also inhibits ice crystal formation, allowing the fish to exist with a lower body temperature than it could have before, because before it gained this antifreeze function, the ice crystals would have burst through its cells, killing it, because I don't know if the audience knows generally, but if your cells get burst, that's bad for you. That's for the audience there, right? Like, you need your cells to be cells instead of just piles of unassociated plasma and lipids and whatnot.
So it sounds like you're saying, okay, so there was a fish that didn't have blood.
Nope, it did have blood, but it didn't have
no.
antifreeze
They could withstand. Yeah, they could withstand this temperature.
Yeah,
It couldn't withstand it before it had this protein.
Right. It could withstand certain temperatures. Like any animal, right, has a range of temperatures that it can exist in.
Right.
And as the temperature cooled around Antarctica, this created a selection pressure, right? So some of these fish had a double copy of certain genes. Like, it's actually not uncommon to have more than one copy of a particular gene. For instance, you as a human have multiple copies for the gene that makes amylase, which is a protein that helps you break down starches. You have one that expresses itself in your salivary gland. You have several that express themselves in your stomach and intestines and whatnot. And they're different copies, right? But they all make amylase. It's the same protein, but you have more than one copy of it.
Right. So that's a copy then.
Right.
Copy of one that already existed.
Right. Exactly. So what happens is this fish gains a new copy of the same blood protein that was originally just used to break down other things, right?
Mm-hmm.
But the copy or the original, doesn't matter, right? One of those two copies that it now has mutated. And the mutation, rather than causing that protein to break down other proteins, instead, something about that protein inhibits the formation of ice crystals, which allows the water in its body, in its cells, to remain liquid at lower temperatures, allowing it to exist in a new range of cold temperatures that it previously could not. That is, if that's not a new function, I don't know what could possibly be a new function.
No, it's a modification of the same.
Yes,
that's
what evolution is. That's the point.
Yeah, but I specifically asked for a new function, new structure, new function.
The original protein did not function in antifreeze. A version of it now exists in a function totally unrelated to its original function. It is definitionally a new function.
Yeah, if this isn't a new function, I can't imagine what would be.
Like, breaking down trypsinogen is not antifreeze, and antifreeze is not breaking down trypsinogen. Those are radically different things, right?
Yeah, honestly, apart from just not wanting to accept that a new function has evolved, I don't know what possible objection you could raise to this.
Yeah, this example is another one that's, it goes deep, right? It's a mechanism that the lame, most of the population is not familiar with blood and temperature. Most of the population
isn't evolutionary biologists.
Well, this example has been around since the late 90s. Like, it's older than me. So,
Jackson, stop bragging about how young you are, you jerk.
I'm also from the late 90s, but this example predates me because I've read the original paper on it. It predates me. So it's not that it's new, like they just came out with this last year. That these examples are in the literature. The thing you have to go through the literature, as Dapper and I do, for we make like debunk videos, and sometimes we just make explanation videos. We go through the scientific literature and look for stuff, and we get deep into it, and so do scientists, and that's why over 99% of them accept common ancestry, because they're aware of these examples and many more, right? That we could go all night with examples like this, but I don't want to hold
You
you
should
for that long.
you should post the link to this one, because I almost
sure.
guarantee it's the way it's being
I'll
described is
be posting the link.
yeah, that'd be great. I'll look at it later,
The
because
way it's being described is what?
I'm hearing that it's the same function that existed. So you saying duplication? That's a duplication of a sequence or a nucleotide that previously existed, and so now that something is being modified within an existing structure. That is not a new structure. That is just a modification.
Okay, so the original protein, the trypsinogen protein. I'm sorry, I've been saying it breaks down trypsinogen. It breaks down trypsin. My bad. The protein itself is called trypsinogen.
Yeah, there you go.
My bad. Sorry, my bad, folks. The
didn't
protein, the trypsinogen, has a different sequence than the antifreeze protein.
Sure.
It has this long, like over 1,000 nucleotide sequence. The only thing that got duplicated from that trypsinogen protein is a 3 amino acid sequence that was then duplicated like 40 times. So the actual sequences of both the original protein and the new one are different. However, that 3 amino acid sequence is derived from the original trypsinogen. Also, for $2, what example would count for Monk Man? We'll answer that in a moment. But do you see what I mean? Right? It came from the original, but it is now fulfilling a different function.
A function that did not previously exist in these organisms.
That function is antifreeze. You're saying that?
Yes, it functions as a
new
biological
functions,
antifreeze. It prevents ice crystals from forming in the cells of this fish.
And you're positive that function did not exist in any other capacity previously?
Yes, there was no other antifreeze gene because first, we can't find it in those fish's closest relatives, which even according to your definitions would count as genetically related. Further, we can tell that this particular gene is actually descended from a gene that had a completely different function, which means that in order for it to be evolved from that, then it evolved from a gene that didn't have that function, right? Or a protein that didn't have that function. Like I got to be honest with you, Monk Man. Like we've had a very friendly conversation. I like you. You seem like a nice guy. This has gone very well. But at this point, it seems like you are being intellectually dishonest. Now, maybe with yourself, right? I'm not accusing you of intentionally lying or anything. But we've already explained that the way evolution works, right, is modifying what comes before. That is the fundamental of how evolution works. It does not work by just new things spontaneously popping out of literally nowhere with no antecedent. That would itself be more like miraculous special creation than it would be like evolution. You've asked for a new function. We've given you an example of a gene that has evolved in very recent times to create a function that did not exist in an organism previously, as evidenced by other organisms as well as that organism itself that are all related even by your own criteria. And your response basically comes down to, but I don't like it though.
Also, I found
that's
the paper.
Extremely frustrating. Do you understand why that does not seem like a good faith response?
It's your interpretation. It's your interpretation that I don't like. The theme of our disagreements tonight has been there's a general rule to something, whether it's mutations or interbreeding, and then you guys find this wild outlier, and that I don't know much about, and I have to do a bunch of research on because it's all these terms and all these definitions, and then you say because I don't immediately accept your interpretation of these outlandish examples, or not outlandish, but unfamiliar. They're in depth, right? They're more in depth than maybe another common example.
Sure.
Because I don't immediately accept them, you say, he's stubborn. He's this, is that. No, come on. I'm a skeptic, right? I'm a skeptic at heart. and that's,
Okay,
So
hold on. I don't believe that you're a skeptic, Monk Man, and here's why, right? What skepticism is not believing claims until sufficient evidence has been provided, right? But here's the problem. Literally hundreds of thousands of papers across more than 100 years have supported universal common ancestry without exception. But you don't accept the actual evidence. Instead, you have an unevidenced conclusion that you have prejudged biology with. And you are sticking to that conclusion not because of the evidence, because let's be honest with ourselves. If the evidence pointed against this, then it would not be the case that essentially everyone who studies it comes to the same conclusion, which is contrary to yours. You're not a skeptic. You are instead refusing to accept the actual evidence because you have a prior conclusion, which, you know what, you're allowed to do, right? I can't tell you you're not allowed to. But what I will say is that is not the behavior of a skeptic. Skeptics accept conclusions for which there is sufficient evidence. To say that there is sufficient evidence to conclude universal common descent for known life on Earth is like the understatement of the millennium, right? There is as much evidence for common descent as there is for the globe Earth. There is as much evidence for common descent as there is for heliocentrism, or for the universal gravitational law, or for like basic like electromagnetic theory. These are or germ theory. These are not like different in levels of confidence in science. There is no more question in science about universal common descent than there is about universal gravitation. So when you say you're a skeptic, I cannot believe you because you have obviously not followed the evidence. Instead, you have come up with, because let's be frank here, right? You couldn't come up with a reason why different families couldn't be related. When presented with novel function, you just kind of hand waved. When presented with examples of why your hybridization thing might not work as well as you did, it's fine to say, I don't know, but you have to realize that that means that you don't have good reasons for using that as a criterion until you do know. So at every step of the way, you have, and I'll say admirably, admitted when you don't know things. But to say that then you're a skeptic with rejecting the claim, because you are rejecting the claim. You're not just simply saying, I don't believe the claim. You reject the claim. When you do that, you're not acting like a skeptic. So please don't call yourself a skeptic when you are demonstrably not a skeptic. You are instead a denier, which is a thing that you're allowed to be, right? I can't tell you you're not allowed to be a denier. You can deny whatever you want. But that's what you're doing.
Science is all about deniers, rejecting claims. That's how every single hypothesis and theory has ever started out.
Science is about skepticism. Scientists are skeptics. Deniers are people who reject the evidence in order to deny obviously true claims. You're in the same category as vaccine quote-unquote skeptics, which are just anti-vaxxers. You're in the same category as globe skeptics, which is just flat earthers. You are not a skeptic. You are an evidence denier. And look,
I'm in the same category as Galileo.
No, you're not.
Denying the consensus.
With Galileo, you are, because Galileo actually did not have sufficient evidence to demonstrate heliocentrism. So
for different reasons.
Yeah, for different reasons. I would actually say that, yeah, you actually are not super far away from Galileo. But you're closer to J.M. Truth, the flat earther, than you are to Galileo.
So I because we have gone way over the 2-hour mark, I will try to bring this down a little bit.
By the way, I'm not saying those as insults, right? Like I still think that like we've had a nice conversation, and I don't dislike you much.
No, this was a very cordial conversation, especially compared to ones that Dapper and I have been in the past. This was a very cordial conversation. I do want to say, however, Monk Man, that definitely none of the claims we've made tonight are honestly outlandish. But what we are saying is, here are resources that you can go and investigate these for yourself. I have a channel, Dapper has a channel, and we link sources to the claims that we make in the description. So if we say like, fossil so-and-so, we'll actually link the paper to that fossil in the description so you can go read about it. And I would, if you go, and a lot of times in video, we'll actually show the paper, we're talking about.
Yeah,
But-
I often at least audibly name the paper and the authors
Right.
and the publication date and the publication venue.
Right.
Oh, perfect. Okay.
So in our videos, you can go and watch the video. You can scroll down to the description, open that up, and see the various papers we've cited. We are perfectly happy to to bear all of our sources for review. But yeah, I do have to agree with that. It's perfectly fine to not know things, but you really do need to do a lot more research into these different claims. Yeah, so that's
Actually,
Uh-huh.
I would be very fascinated to have another conversation.
Absolutely.
After you've looked into things like the hamster test and like ice fish antifreeze proteins and
Yeah,
things
definitely.
like embryological development of the spine from a notochord. Like, and look, I'm going to be honest with you. Like, that's a lot of homework, right?
Right.
Yeah.
That's not like an afternoon worth of homework. This is
The
like
two of us have been like
months
reading
worth.
technical papers about biology for years at this point.
Right?
Yeah, we've
Years
both
and
been
years
doing
and
it for
years.
years.
So, like, I don't expect that, like, tomorrow you're going to come back and be like, I am now an expert on the hamster test. Like, of course not, right? But,
Right.
like, in a few months, it would be fascinating if you looked into these things and you familiarize yourself with the technical literature. Maybe you familiarize yourself with, like, the techniques behind, like, cladistics and phylogenetics. You familiarize yourself perhaps with what would be really fascinating would be, like, if you could find what science would call closer related groups, like we use with the example of Procyonidae and Ursidae. And you could look into, like, the technical literature about their genetics and anatomy and physiology and whatnot, and synthesize all that into a more specifically informed counter to common ancestry. Because, I mean, quite frankly, it's almost unheard of that we actually get to talk to someone who's a creationist who's familiar with, well, technical literature at all, ultimately.
Yeah.
Because the creationists who I find who are familiar with technical literature, I also find to be blatantly dishonest, like doing things like saying, oh, hey, there are no fractures in this rock formation, and here's the evidence, and show a picture. And there are curiously people positioned all throughout the picture in front of the rock formation. And then you check the USGS picture of the same rock formation, and oh, lo and behold, there are fractures exactly where the people are standing. That's the kind of dishonesty I'm talking about with creationists who I know of who are familiar with the technical literature.
Or they will cite a paper and say, aha, it says this. Then you read the paper and it's like, no, it says the exact opposite of that.
Yeah.
Or something like that.
So, like,
that happens.
I would love to have a conversation with a creationist who has familiarized him or herself with the technical literature and who is being honest about it. And I think that you have the opportunity to be that guy if you go into it. Although I will say, I suspect, but cannot prove, that the reason that all the creationists I'm aware of who are actually very familiar with the actual literature, with maybe one or two exceptions, are, instead of being honest with it, just being obviously dishonest with it. And I suspect that the reason is because creationism just isn't true. So there's no way to be both honest and well-informed about the topics that relate to it. I suspect that that's the case. However, I would love to be proven wrong, because here's the thing, right? I'm a former young Earth creationist, and if you could demonstrate to me that that's true, that would be great. Because, I mean, that would answer a whole lot of philosophical and theological problems and questions that I would love to have easy answers to, right? Like, if you could show, like, hey, the one true religion set has now been reduced to, like, these few options, well, that would be great. That'd be amazing. I'd love that. And if you could do it on the basis of, like, demonstrable science, that'd be pretty cool. So, like, I'm not someone who doesn't want creationism to be true. I'm someone who reluctantly came to the conclusion that there just isn't any evidence in support of it, and instead there's mountains of evidence to the contrary.
Yeah. So, in closing, I do want to say thank you again, Monk Man, for coming on and discussing this with us. We did a very pleasant conversation.
Thanks for watching, but before you go, I just want to say a special thank you to my channel members and my patrons, especially those pledging $20 or above. Bob Knob, Work in Progress, Bent Hovend, Tapioca Weasel, Denny5252, Ian Chen, Landon Knoll, Mabdy Babdy, San, Sphincter of Doom, The Venerable Veed, Eleron Teller, and Pat L. Dennis. As you probably know, YouTube is a very volatile platform, and from month to month my income on the channel can swing wildly. But the people you see on screen are directly supporting so that I don't have to worry too much about that, and the channel can keep going as it is, and perhaps even improving. If you'd like to join this team, there are links below to join the channel or in the description to join the Patreon. The Patreon starts as low as $1 a month, and the channel memberships start as low as $1.99 a month. On Patreon, you can even get a discount for pledging annually. If you do decide to pledge, you'll get access to an exclusive Discord server just for channel supporters, as well as early access to all of my pre-recorded videos, often up to 2 months in advance. Higher tiers will unlock even better perks. Now, if the annual or monthly pledge isn't right for you, but you still want to support the channel, there is a merch store down in the description, as well as an Amazon wishlist, just for me. And if financial support isn't something you can or want to do, then if you still want to help out the channel, please just like and share these videos and make sure you comment on them. It really helps the channel out. Thanks again for watching.
How